Last but not least, we will have Xuewei Wang, who I believe is a postdoc in Fengquan Zhou's lab at Johns Hopkins University, and he's going to talk about also on the subject of axon regeneration. So we start with that and end with that. Go ahead, Xuewei.
Sorry, I was muted.
We can hear you.
Yeah. Can everybody see my screen?
Yes.
Okay, perfect. All right, thanks for the warm introduction. So my name is Xuewei Wang, and I'm a research associate, aka a senior postdoc at Johns Hopkins University. I'm really honored today to speak at this year's SCBA DC-Baltimore chapter symposium, especially along with all these exciting talks. So today I'll be talking about our recent study on regenerating axons in the central nervous system by epigenetic rejuvenation. So current clinical treatment for neurodegenerative diseases and neuroinjuries falls short of success, primarily because neurons in the central nervous system lose their axon regeneration ability as they mature. On the other hand, neurons in the peripheral nerve system can spontaneously regenerate axons upon axonal injury by initiating a regenerative response. So over the years, a number of studies have implied that the loss of axon regeneration ability of CNS neurons and this injury-triggered regenerative response of PNS neurons largely involve changes in the chromatin structure and accessibility. Our goal is to understand what roles epigenetic regulation plays in the developmental decline of axon regeneration ability of CNS neurons, and investigate if modulating epigenetic regulators of chromatin structure and accessibility can enhance axon regeneration in the central nervous system. And here in this study, we investigate the role of an epigenetic regulator, EZH2, in mammalian axon regeneration. So first of all, what is EZH2? EZH2 is the catalytic subunit of the polycomb repressive complex 2. It functions as a histone methyltransferase to add methyl groups to lysine 27 of histone 3, or H3K27 for short. Trimethylation of H3K27 is a heterochromatic histone modification, which means it makes the chromatin more condensed to suppress gene transcription. In recent years, functions unrelated to histone methylation have been reported for EZH2, indicating it is a multifaceted molecule. So there are two major reasons we're interested in EZH2. First, we found EZH2 protein level is developmentally downregulated in both the PNS and the CNS, indicating it is an important factor for nervous system development. And second, which is more important, we found EZH2 level in PNS neurons can be upregulated by axonal injury. So here we performed a sciatic nerve transaction, and 3 days after the transaction, we found a sharp increase of EZH2 in lumbar 4 and 5 dorsal root ganglia, or DRGs for short. As I mentioned earlier, PNS neurons are capable of spontaneous axon regeneration. These observations led us to think that this injury-triggered upregulation of EZH2 might be related to the axon regeneration function of DRG neurons. So here we use an in vivo axon regeneration model to test this hypothesis. We electroporated siRNA against EZH2 in DRGs to knock down its level in lumbar 4 and 5 DRGs and performed an sciatic nerve crush. GFP plasmid was co-electroporated to label the axons. 3 days after the nerve crush, we found the average length of regenerated axons was shortened by about 20% compared to the control group. So to exclude the possibility that this was caused by off-target effects of the siRNA we used, we obtained EZH2 flux mice and electroporated Cre plasmid to knock out EZH2 level in DRGs, and also performed a sciatic nerve crush. And again, the results show that EZH2 loss of function impaired axon regeneration ability of DRG neurons in vivo. So to further exclude the possibility that this was caused by EZH2 loss of function in non-neuronal cells in DRG, we crossed advenin Cre mice with EZH2 flux mice to obtain a sensory neuron-specific EZH2 knockout mouse line. And again, for the third time, we showed that EZH2 knockout impaired axon regeneration of DRG neurons in vivo. So since the upregulation of EZH2 in DRG neurons is necessary for their normal axon regeneration function, we wondered if forced overexpression of EZH2 in non-regenerative adult CNS neurons can also enhance their axon regeneration ability. So to address this question, we used the optic nerve crush and regeneration model. But before getting into the details of that experiment, I want to give a little more background of EZH2. Many mutant forms of EZH2 have been characterized in human cancer studies. More than a decade ago, a leukemia study found that if the 731st amino acid of human EZH2, which is a tyrosine, is mutated to an aspartic acid, the methyltransferase function of EZH2 is completely lost. This is because the tyrosine 731 of human EZH2, or the equivalent tyrosine 726 of mouse EZH2, is located in the core of the catalytic domain of this molecule. So here we overexpress wild-type EZH2, or the mutant form of EZH2, in retinal ganglion cells, or RGCs for short, by intravitreal injection of AV2 vectors, and confirmed by immunostaining that the overexpression of wild-type EZH2 significantly increased the trimethylation level of H3K27 in RGCs, whereas the overexpression of this mutant form had no effect at all. So we performed an optic nerve crush after the overexpression of wild-type EZH2 or the mutant form, and 2 weeks after the nerve crush, we found EZH2 overexpression induced many RGCs to regenerate axons. Some of them could grow for more than 1,500 microns in this 2-week period. To our surprise, this catalytic dead form of EZH2 was still able to modestly induce optic nerve regeneration, indicating EZH2 promotes axon regeneration via both histone methylation independent and dependent pathways. To gain mechanistic insights into how EZH2 supports axon regeneration, we performed RNA sequencing of control and EZH2 overexpressing RGCs. Hundreds of differentially expressed genes were identified between these two conditions, and among them, we were particularly interested in 3 groups of genes. Given that EZH2 primarily functions as a gene transcription suppressor, we first focused on genes that were downregulated by EZH2 overexpression. The first group is comprised of synaptic transmission-associated genes that mainly encode receptors, channels, and transporters. Since synaptic transmission is a fundamental function of mature neurons, the fact that EZH2 suppresses the expression of these genes indicates EZH2 overexpression could rejuvenate mature RGCs. To verify this idea, we performed RNA sequencing of postnatal day 1 and postnatal day 14 RGCs, where P1 represents young RGCs with strong axon growth ability, whereas P14 represents RGCs with poor axon growth ability. And we found that these genes are usually expressed at low levels on P1, but are generally highly expressed on P14. In other words, the transcription of these genes increases during RGC maturation, but the overexpression of EZH2 in mature RGCs reverses this increase, indicating EZH2 supports axon regeneration by rejuvenating mature neurons to empower them with stronger axon growth ability. To determine if these synaptic transmission genes functionally act downstream of EZH2 to regulate axon regeneration, we selected SLC6813, which encodes GABA transporter 2, and was one of the genes most significantly downregulated by EZH2 overexpression in RGCs to study. We first confirmed by qPCR that its RNA level was decreased by EZH2 overexpression in RGCs. In addition, we used the cut and tack method, which is similar to CHIP, but can be done with fewer cells to show that the promoter region of SLC6813 is bound by H3K27 trimethylation, indicating that it can be directly regulated by EZH2 at the transcriptional level by histone methylation. When we overexpress it along with EZH2 in RGCs, the optic nerve regeneration induced by EZH2 overexpression was partially blocked, indicating EZH2 supports axon regeneration partially by transcriptionally suppressing GABA transporters. The second group is another group of genes downregulated by EZH2 overexpression, and they encode some major CNS axon growth inhibitors. OMGP is one of the three major myelin-associated inhibitors that act to inhibit axon regeneration. Tenascin R is an extracellular matrix molecule that was shown to be a repulsive axon guidance cue during development and an inhibitor of optic nerve regeneration. Although LINGO-3 has not been directly linked to axon regeneration, its paralog LINGO-1 encodes a critical component of the noble receptor complex, which can be activated by myelin-associated inhibitors and CSPGs to block axon growth. In addition, LINGO family receptors have been shown to form heteromers with one another in the mouse brain, suggesting functional overlap between the paralogs. Afferent receptors are chemorepellent axon guidance cues that can be activated by their ligands, afferents, to induce growth cone collapse. It is widely accepted that CNS axon growth inhibitors can be categorized into three classes, which are myelin-associated inhibitors, CSPGs, and repulsive axon guidance cues. Our results show that EZH2 overexpression suppressed the transcription of inhibitors or their receptors associated with all three classes, indicating EZH2 is a master suppressor of CNS axon growth inhibitory signaling. Next, we tested if the downregulation of OMGP and LINGO-3 contributed to the optic nerve regeneration induced by EZH2 overexpression. We found that overexpression of OMGP or LINGO-3 significantly blocked the optic nerve regeneration induced by EZH2 overexpression. And the quantification results here show that only at 250 micron away from the crutch site can we observe more regenerating axons in the two co-overexpression groups than in the control group. So collectively, these results demonstrate EZH2 supports optic nerve regeneration by transcriptionally silencing major axon regeneration inhibitory signaling. The third group is a group of genes upregulated by EZH2 overexpression, and is comprised of many well-known positive regulators of axon regeneration. Among them, ATF3, JUN, GAT45G, SPR1A are some of the first identified regeneration-associated genes. Others, including MYC, SOX11, osteopontin, IGF-1, thrombospondin-1, and Dextin-1, have been proven to promote optic nerve regeneration. Osteopontin has been shown to selectively promote axon regeneration of certain RGC subtypes. In the meanwhile, increased osteopontin expression has been shown to underlie the enhanced tissue repair induced by WFDC1 knockout. WFDC1 is a gene known as a wound repair inhibitor. Interestingly, the RNA level of WFDC1 was also decreased by EZH2 in RGCs in our RNA-seq results, indicating it might regulate EZH2-induced optic nerve regeneration via osteopontin pathway.
13
and
minutes,
indeed,
Xuewei.
all right, thank you. And indeed, we found that overexpression of WFDC1 completely blocked EZH2-induced optic nerve regeneration, indicating its downregulation is necessary for the EZH2-promoted axon regeneration. Since EZH2 primarily functions as a gene transcription suppressor, these results provide a potential mechanism of how EZH2 indirectly upregulates axon regeneration enhancing factors. So to summarize, our study found that EZH2 supports mammalian axon regeneration via rejuvenating mature neurons, suppressing multiple classes of axon growth inhibitors, and activating various axon growth programs. And we also demonstrated that modulating chromatin structure and accessibility is a promising strategy to enhance axon regeneration in the central nervous system. Yeah, I want to thank the members in the Zhou lab who participated in this study, particularly my mentor, Dr. Fengquan Zhou, who's going to speak at the career panel discussion in a few minutes. Also, special thanks to Dr. Mingwen Hu from the Chen Lab, who did massive analysis work on our sequencing data. I also want to take the time to acknowledge the core facilities at Hopkins and also the funding agencies that support me and Dr. Fengquan Zhou. And with that, I'm going to stop here and take questions. Thank you.
Thank you, Xuewei. That's a very interesting talk, and it's open for questions. We have one from Yihong. Is there any advantage to lose regeneration in CNS? That's a big question.
Oh, yes. Right. This is a really great question. So if evolutionarily, low-level animals are capable of axon regeneration, but mammals like human and mice are not able to regenerate axons in the central nervous system. I guess, I can't say for sure, but I guess it's a way to save energies for the central nervous system, as a previous talk has shown that a CNS neuron, consumes a lot of ATP in a single second, right? So, yeah, that's. I hope that answers the question.
Maybe it's not worthwhile to save the brain damage that. We have the next question from Xin. Yeah. Go ahead, Xin.
Hey, Xuewei, it's a great talk. I just wonder, do you know what triggers the upregulation of EZH2 after injury? And does that recapitulate what's going on in earlier development?
Oh, yeah, I think that is our next study. So actually, we don't know, because we just studied the downstream mechanisms of EZH2 overexpression, and we only noticed that EZH2 was dramatically elevated by the axonal injury, but really we don't know why, what mechanism caused the elevation of EZH2 in those DRG neurons.
Yeah, do you know if this upregulation is at the RNA level or protein level, or both?
Oh, yeah, I think it's both. So I also have the data of RNA increase of EZH2 in DRG neurons, but I just didn't include it in the slides, right? So both the RNA level and protein levels were upregulated, right?
Yeah, thank you.
So related to that, how do you get to EZH2? What is it, through a screening or educated guess? How do you get to that?
Oh yes, through screening. So we screened some epigenetic regulators of chromatin structure and accessibility, and find this one was really upregulated by this axonal injury.
And is it the only one, or there are more modifier or regulator of epigenetics?
Yeah. Well, I mean, there's a lot, but this one is one of those most prominent ones. So that's why we followed on this one to study it.
Thank you. Someone, yeah, go ahead.
Yeah, very similar to Xian. When it says you see the both RNA level and the protein level increase, do you understand, is that due to transcription or RNA stability to see the RNA increase?
For that, I don't know. I mean, it can be both transcriptional or post-transcriptional, but I don't know it.
One last question just popped in. Does regeneration ability also decrease in old CNS cells? Is it age-related?
Yeah, let me see. So basically, when the initial axon growth of the neurons competes, the axon regeneration level, the axon regeneration ability of the neurons just is pretty much lost. Well, I would say it continues to lose during aging, but the most critical change happens shortly after the initial axon growth completes.
Okay, so that pretty much concludes the session. It's a very informative, intriguing session, and we wanted to thank all the speakers and the audience. And now